Alright class, so here is the rest of our DNA review sheet for you. Let me get my pen up. We'll do thin red. Red's a good color. Okay, so the next question, we finished up with what is covalent bonds and where does it happen at in DNA? And there we go. We were right here and we talked about a covalent bond being where they share electrons. And in the DNA where it happens is the backbone. And I drew the picture of the backbone of the DNA. DNA looks like this. And the backbone of the DNA is this part. Okay, so it's the outside of the latter part. Now, hydrogen bonding. Hydrogen bonding is when we have, say we have our water molecule, our Mickey Mouse ears, remember those? We have two water molecules. So we have this negative side of the oxygen and this negative side, and we have this positive side of the hydrogens. This positive right here is attracted to the negative of the other water molecule's oxygen. So this right here is the hydrogen bond. It's the attraction between two molecules. And hydrogen bonding is really not a bond that happens. It's more an attractive force between these two molecules. So where does it happen at in DNA? It actually happens between the nucleotides. So we have our DNA. I'm going to zoom in a little bit. So here's our nucleotides. We have our A's and our T's that are attracted to each other, and we have our C's and our G's. This right in between here, that is our hydrogen bond. That is our hydrogen bond. Okay, and that is important that you know that's where the hydrogen bond are. Out here is a covalent bond. That's where they're sharing their electrons. Next part. What happens during DNA replication? What is the main goal? The main goal of DNA replication is to get a second strand that is identical, which is identical. That is our main goal. Also, know the enzymes, ASE endings, and their functions that play a role in DNA replication. So we have two of them. We have DNA polymerase and it's going to bring in the nucleotides and proofread and then we have our helicase. And our helicase, remember, that is going to unzip the DNA. Where does DNA replication take place, and why is it important that it happens during this time? Okay, so DNA replication is going to take place in the nucleus for eukaryotes. And it happens in the S phase. And that is the synthesis phase of the cell cycle. S phase. And it needs to take place in this time because it won't, when the cell starts to divide in the cell cycle, that way both cells get a strand of DNA. What is the difference between DNA and RNA? Well, DNA is deoxyribonucleic acid, so it has oxygen in it. RNA does not have oxygen. Also, another difference is the nucleotide for RNA is uracil. Uracil. And DNA, it's thymine. Those two actually change. So then it's A and U's go together for RNA, and for DNA, it's going to be A's and T's. Draw a picture of replication. Okay, so our picture, we're going to have our DNA strand. Make it go bigger right here. Okay, so right here we're gonna have our helicase. This is our helicase. Maybe I'll change colors to green. Here's our helicase. Helicase. Unwinds DNA. Then our next picture, we're going to have our DNA polymerase. That's going to be blue. DNA polymerase is going to bring in free-floating nucleotides. So DNA polymerase brings in nucleotides, bonds them together, and proofreads. Sorry. And then lastly, we have two new strands of DNA. Two new strands. Okay, if you have the sequence AG da 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 da, what is the corresponding DNA strand? So what is the other pair? So remember, A's goes with T's, G's goes with C's. So if we have A, we have to have a T, then C, then G, then A, C, C, A, T, C, G, C, A, T, G, A would be the corresponding DNA. So A's and T's go together, C's and G's go together, and so on and so forth. Next question. Describe biotechnology and how has it brought us to the discoveries of DNA. Predict how it will help in the future. Biotechnology is bringing biology and technology together. It has brought the discovery of DNA to happen. If we did not have biotechnology, we would not have discovered DNA. Remember, we have the X-ray picture. We also have the microscopes. We have computers now that give us animations of what things actually look like that take place in the body. So we have computers. If we did not have these things, there's no way that we would even know what DNA looks like, or we would even know what cells look like. Especially when we start talking about transcription and translation, there's no way that we would even depict what those things look like either. So there is the rest of the review for you. If you have any questions, please come in.